Okay, good morning students. Today we are going to discuss fourth experiment of class 12 biology. Class 12 biology, fourth experiment or fourth practical. In this practical, what is the name of the experiment or practical? It is to study and identify. The practical is to study and identify the stages of gametogenesis, that is spermatogenesis and oogenesis. Stages of gametogenesis, that is spermatogenesis and oogenesis. You know there are two types of gametogenesis. So the process of formation of sperm or spermatozoa, this is called spermatogenesis. Site is testis, seminiferous tubules of testis. And oogenesis, this is the process of formation of ova or egg or ovum or ova. This is called oogenesis. This takes place, this occurs in the ovary or ovarian follicles. Okay. Now, first of all, let me discuss spermatogenesis. Requirement: we require permanent slide of TS testis and TS ovary. We need a compound microscope for study. Procedure: first of all, we have to take a permanent slide of TS testis, and this slide of mammal, TS testis of mammal we have to study, TS testis of mammal, and then this slide is placed on the stage of the compound microscope and focused. After focusing, we can visualize, we can see the TS testis, we can see the diagram, we can see the picture, pic, and now comes the observation. When we see the pic, what we observe? We can see many rounded, when we cut the TS of testis, in TS testis, we see many rounded structures called seminiferous tubules. So seminiferous tubules, these are the sites of spermatogenesis. These are the structures in which spermatogenesis takes place. Okay? Now there are a few very small oval or rounded cells in the space between the seminiferous tubules. These small cells, rounded cells, oval cells are called cells of Leydig. And the cells of Leydig is endocrine in nature, and this what secretes chief male sex hormone, chief androgen, that is male sex hormone, testosterone. Now let me come to the diagram. You have to write the procedure, and you have to draw the level diagram also on the white sheet. Now this is the TS testis. We can see rounded structures. These are the seminiferous tubules. In the seminiferous tubules, you can find this is lined by epithelium. This is the called germinal epithelium. The cells are diploid. The cells of diploid cells of the germinal epithelium undergo meiosis to produce the haploid cells. And these haploid cells called spermatids enter the Sertoli cells. There are some pillar-like cells. These pillar-like cells called Sertoli cells. So Sertoli cells are the nerve cells. These are the cells where or in which sperms develop. Okay? The spermatids, haploid cells called spermatids, enter the Sertoli cells. In the Sertoli cell, spermatids are transformed into sperm. So during the transformation, during the development, the Sertoli cells, these pink colored Sertoli cells, these pillar-like cells called nerve cells, these cells nourish the developing sperms. And after development, the sperms are released into the luminal space. So these are the sperms. You can mark over here. These are the sperm. Sperms. So this is about the spermatogenesis. Okay. Friends, let me discuss the second part of gametogenesis, that is oogenesis. What is oogenesis? The process of formation of ova or ovum in the ovary or in the ovarian follicle is called oogenesis. So ova is the female gamete, ovum is the female gamete, that develops in the ovarian follicles of the ovaries. So what we have to take? So 1 minute. 1 minute. Second part of what gametogenesis, we are going to discuss oogenesis. What is oogenesis? The process of formation of ovum or ova in the ovary is called oogenesis. Now what things we require? We require a permanent slide of TS ovary of mammal, a microscope, compound microscope. Similarly, the slide is what is placed on the stage of the microscope, compound microscope, and focused. After focusing, let me discuss the observation. What we observe, what we find, what we see in the slide. In the slide, we can see the structure like this. The ovary is album shaped. It is almost album shaped structure. This is called ovary, TS ovary. Okay. Now in this ovary, you can see ovary is lined by a single layer of epithelium, single layer of epithelial cells or epithelium. This is called germinal epithelium or oogonial cells. So this we can mark over here. We can mark germinal epithelium. This is the germinal epithelium. So these cells are diploid. These cells first of all divide by mitosis and give rise to large number of cells, and these what cells, diploid cells, form follicle called ovarian follicles. So we can see a number of follicles in the ovary. TS ovary, we can see a number of follicles at different stages of development. Follicle may be primordial, may be primary follicle, secondary follicle, tertiary follicle. Okay, and the matured tertiary follicle is called Graafian follicle. We can find this one. This one is called the tertiary follicle. Tertiary follicle formation has taken place. So this is the follicle, and inside the follicle you can find the ovum. This is the ovum developing inside the follicle. This is the ovum. Ovum developing inside the follicle. Now the follicle has developed, the ovum has developed. Finally, the follicle ruptures on 40th day of the menstrual cycle, and the ovum is released. Ovum has been released into the body cavity. This is called ovulation. And after ovulation, within a day or 1 day, approx 24 hours or 30 hours, this ruptured follicle, this changes into golden yellow structure, endocrine structure called corpus luteum. And you know that this corpus luteum secretes hormone progesterone. Progesterone is the very important hormone, female sex hormone. This is secreted by the corpus luteum. So corpus luteum is endocrine in nature. And this is the ovum which has been released. Now this ovum will go into the fallopian tube where fertilization takes place. So these are the follicles at different stages of development. This outer part of the ovary is called cortex, and the inner part of the ovary is called medulla. So ovarian follicles are present in the outer part, that is cortical part of the ovary, and this medulla, the inner part is called medulla. This is supplied with, in the medullary region or inner part, ovarian follicles are absent. So this part is supplied with blood vessels. Blood vessels come to this area, this way, and the ovary is attached to the body wall से attached रहता है, ठीक है? So this is the ovary. This is the TS of ovary. You can see the germinal epithelium, ovarian follicles, and this is the corpus luteum, ovum, corpus luteum. Ovarian follicles at different stages, germinal epithelium, medulla region, blood vessels, and this outer region called cortical region or cortex. So this is the diagram. This structure you will see when you focus the slide on the microscope. You will find this structure. This is the TS of ovary of man. Okay, the theory part, you have to write the points, procedure, materials, things required, observation, and then you have to draw the level diagram also. Okay, that's all. Okay. Now students, you can see this one. This is the slide of TS testes. I am going to focus the slide of TS testes. Similarly, we can focus the slide of TS ovary also. No problem. So this is the compound microscope. This we have taken compound microscope. This compound microscope, its mirror should be towards the window side so that it may get the sufficient light. So this is focused towards the window side. So light is coming from the window side, and now we have to focus this. We have to place this slide on the stage of the microscope. So this is the stage. This is the stage of the microscope. So I have focused the slide, and then we can fix the slide with the help of stage clip. There are clips called stage clips. With the help of these clips, we can set the slide. We can fix the slide. Now, with the help of broad adjustment screw, we can focus it. Once it has been focused with the help of broad adjustment screw, then we can use the fine adjustment screw. With this we can make the fine adjustment according to our vision or eyesight. Now it's clear, we can visualize, we can see the what? We can see the many seminiferous tubules, and in the seminiferous tubules we can see the Sertoli cells, we can see the germinal cells, we can see the Sertoli cells, and we can see very fine minute sperm cells. Okay, so this is the procedure of the practical. So in the practical spotting, this question is asked in spotting. In spotting, the slide will be already set. What you have to do, you have to come to the place, spot number, and you have to see the slide, identify the slide, and then you have to draw the diagram, level it, and identify that the given slide is whether TS testis or TS ovary. So this is the procedure. So this is the question of spotting. This experiment, this will be in spotting item. ठीक है, okay.